Hello, this is Sanghavi, Assistant Professor, Department of Pharmaceutical Chemistry, LJ Institute of Pharmacy. Today, we are going to talk about three important aspects from biochemistry segment, that is introduction to lipid, amino acid and protein. Fine, let's start. When I talk about three important aspects, protein, amino acids and lipid, they are essential biomolecules. Let me start with lipid. What are lipids? Lipids are generally called as an organic substance, insoluble in water, soluble in organic solvent, potentially related to the fatty acids. Generally, we say that fatty acid esters are called as a lipid. But very important aspect is they are utilized by the living organism. Classification of lipid, if I say, mainly three categories: simple lipid, compound lipid and derived lipid. Simple lipid means what? They are fatty acid esters. The examples are given: fats and oil, and of course the second example is wax. If I talk about compound lipid, of course they are fatty acid esters, but additional group is involved. In case of phospholipid, phosphoric acid is there. In case of glycolipids, sugar moiety is there. So they are called as a compound lipid. If I talk about derived lipid, of course they are as a part of hydrolytic product. Either they are called as a fatty acids, they are called as a alcohol, what you can say glycerol. Glycerol is one of the most important alcohol. Fine. Sterols and certain isoprenoids, that is terpenoids and carotenoids. When I talk about the derived lipid, because it plays very important role. The first one is essential fatty acid. The word itself suggests essential means these fatty acids are not synthesized by the human body. It has to be supplied through the diet. Classical example, if I talk about, is polyunsaturated fatty acids. Two important one, that is linolenic acids and linoleic acid. The nomenclature says everything. If you write in form of a numbers, the first uh, number represents total number of carbon. The second one is how many double bonds are there, and third and fourth one is at which position double bonds are there. So nomenclature is very important. A second is triacylglycerol. The word itself suggests it is what you can say esters of glycerols with fatty acids. All the hydroxyl group from the glycerols are esterified with the you can say carboxylic acids. Fine. Now, when I talk about the second aspect, that is a phospholipid, a complex lipid. Fine. So again, why? Because fatty acid esters are always there, but additional group is definitely involved. In case of phospholipid, it is a phosphoric acid. Not only phosphoric acid, but nitrogenous base and alcohol is always there. Again, classical example is glycerophospholipids. Categories, second category is sphingophospholipids. The difference is glycerol is alcohol in case of first category. Second category is sphingosine is alcohol in that particular phospholipid. Very important part. You can see the diagram of the lipids, phospholipids, right? The diagram itself, this kit plays essential role as far as cell membrane is concerned, because composition of cell membrane itself suggests that it is having phospholipid bilayers. We definitely see. Fine. Again, but it structurally, if I talk about two important aspect, non-polar chain, what you can say tail aspect, long hydrocarbon chain is there, and polar aspect, it means certain esterified group is there. That is why non-polar portion is more. So of course, it is soluble in organic solvent. Fine. Now this is again very important aspect. You can say application point of view, as far as the nerve cell is concerned, right? The structural diagram of nerve cells concerned, marinate sheath, it is having what you can say lipid bilayer. Fine. Now second aspect biomolecule is all about protein and amino acids. We generally say that amino acids are monomer of protein. Proteins are definitely polymer of amino acids. But before we start with the protein and amino acids, we have to be very clear about amino acid first. Amino acids, we all know very well, they are organic one, nitrogen containing compounds, right? And many amino acids joined by the peptide bonds, definitely proteins are formed. It is derived from the Greek word proteos. It means holding the first place. Means it is always there as far as living organism is concerned. Fine. Now, functions of protein we all know very well, because As far as hair is concerned, again it is having protein. Blood is concerned, thousands of proteins are involved in the blood. Bone is concerned, muscles are concerned. So structural aspect, if I say, diversified functions are there. Structural functions, it means it plays very important role, responsible for structure and strength of body. Dynamic functions, it means there are varieties of diversified functions that the protein in which protein is involved. Antibody, again it is a protein, that is immunoglobulin. Receptors, storage protein, right? So functional aspect, if I say, structural aspect of functions and dynamic aspect of functions, that we have to remember. Amino acids, we all know very well. The organic one, nitrogen is definitely there. NH2 group is involved and COOH group is involved. So amino acids are having two essential functional groups, COOH group and of course NH2 group is there. Both are attached with the same carbon. You can definitely say. We all know very well, carbon has 4 valency, 2 are occupied. Now one carbon is attached with hydrogen and another part is called as a R group. Based on that R group, amino acids are definitely classified. As far as classification of amino acid is concerned, we all know very well there are certain criteria based on structure, chemical nature, polarity, nutritional requirements, and metabolic fate. But structure-based classification is very, very important classification. We all know very well. There are 5-6 categories. Amino acids with aliphatic side chain. The R group is having aliphatic side chain. It is symbolized as a red in the structure. Amino acids containing hydroxyl group, amino acids containing what you can say sulfur containing amino acids, acidic group is there, basic groups are there. These are the changes takes place in the side chain. In case of aromatic ring system is definitely involved. So what I mean to say, structure based classification is very, very important. Based on polarity, we all know very well non-polar and polar. But again, as far as general amino acid structure is concerned, it is always a polar. So this classification we have to give based on the R group. If R group is alkyl, is having only alkyl chain, the amino acids are called as a non-polar. If R group is having certain NH2 COOH group is there, definitely they are considered as a polar. Fine. When I talk about nutritional, we all know very well certain amino acids are synthesized in the body, certain are not. So definitely if I talk about nutritional classification, essential amino acids and non-essential amino acids. 10 amino acids are essential and are non-essential. It means they are synthesized in the body. Now, when I talk about the amino acid classification based on metabolic fate, again, 3 categories are there: glycogenic, ketogenic, and both glycogenic and ketogenic. Because these amino acids can serve as a precursors for the formation of glucose or glycogen, and hence it is called as a glycogenic. In case of ketogenic, of course, the fat molecules are synthesized from these amino acids. When we talk about properties, we all know very well. Basic properties, we all know very well. Solubility means being a polar nature. Of course, they are soluble in polar solvent like water. If I talk about melting point, generally melting points are high. Taste, optical property. This is very important. Except glycine, all amino acids are considered as a optical active. But one of the essential feature of amino acid property, that is it forms a zwitterion. It forms a zwitterion, and at which pH it forms a zwitterion, it is called as its isoelectric pH, where it is having minimum solubility in the water. If I talk about chemical property, both the functional groups are there, acidic and basic. So definitely due to a carboxylic acid group, it react with the what you can say certain reactions of carboxylic acids we just have to remember, right? So if I talk about carboxylic acid group, it forms a salt with base, it forms a ester with the alcohol, like that. Fine. Decarboxylation is again reactions of carboxylic acid group. Fine. If I talk about the another aspect, due to NH2 group, of course, being a basic group, it forms a salt with acid. So certain fundamental properties that we have to remember as far as chemical functional group is concerned. Now, when I talk about identification of amino acids, Nain-Hydrin reaction is the only one reactions by which we can identify that the sample is having amino acids. Fine. Now, amino acids gives certain color reactions. Practical aspect, if we see which amino acid is present based on the what you can say name of test, we have to perform certain test. For an example, biuret Millen's test. So certain amino acids is having phenolic group. It gives only positive that particular test. So when amino acid sample is confirmed, you have to approach which amino acid is given, and that is why you have to perform this all particular test. This is a practical aspect. Now, when I talk about protein, more or less similar property with respect to solubility, molecular weight. Yeah, molecular weight is higher as compared to amino acids because now it is a polymer. So many amino acids are definitely there. Isoelectric point, again the same what we have discussed in case of amino acids. But remember one thing, we have to remember at which protein is having isoelectric pH 4.6, 1.1. This is important because in entrance examination point of view, they ask. Fine. We all know very well protein gives certain precipitation type of reactions because what you can say, there are polar groups, and because of hydration of polar group is concerned, it gives precipitation reaction. As far as the main identification test is concerned, amino acids, that was nitrogen I discussed. In case of protein, that is biuret test, right? This is very important aspect of the protein identification panel. But all the protein gives biuret test positive. A single amino acids, that is a histidine, it is the being amino acids, it gives biuret test positive. This is all about practical aspect. Fine, right? It forms what you can say, how biurets are formed. Two molecules of urea, when you hit at high temperature, that is 180 degree, biuret is formed. It forms a complex with the copper ion that gives purple color, and hence we can identify that this sample is protein. Fine. Varieties of classification categories are there: structural protein, enzymatic protein, transport protein. Everything as far as living organism is concerned, we all know very well, it is made up of proteins, right? And definitely in diet, we have to take more protein containing diet, that we all know very well, because you see the functions of proteins are many. Every aspect you can see: storage, genetic, contractile protein, hormonal, transport. Every aspect protein plays very important role. Fine. Based on solubility, again a simple protein, conjugated proteins, and derived protein, just what we have discussed in case of lipid, right? Simple, conjugated, and derived. Just like that, simple amino acid residues, simple protein, conjugated protein, additional group is definitely involved. And if I talk about derived protein, if proteins are getting hydrolysis, undergo hydrolysis, it is converted into peptides, right? So they are called as a derived protein. This is again one more view by which you can remember the classification: derived one, primary and secondary. Proteose, peptones, they are derived protein. Fine. Now, when I talk about nutritional classification, same as well. Complete protein, incomplete protein. It means all the essential amino acids are definitely there. We always say it is a complete protein because structure of protein, ultimately they are amino acids. Incomplete protein means one or more lacking of the amino acids, and hence we surely say that it is an incomplete protein. Now, when I talk about structure of protein, four aspects that we have to remember: primary structure, secondary structure, tertiary, and quaternary. Primary, it means what? Simple sequence of amino acids. Secondary, it means what? Alpha helix and beta pleated sheet, right? Tertiary means three-dimensional structure of the protein. And if I talk about quaternary structure, it means certain proteins which are composed of two or more polypeptide chain, referred to as a quaternary structure of protein. Let me show you the figure. Now, this is the primary one, simple residue, beta pleated sheet, and what you can say, alpha helix. Again, if those helix are in a 3D view, definitely it consists of tertiary structure of protein. And quaternary structure of protein, if I say, it is always a complex one. More than two polypeptide chains are there. Primary, simple residues of amino acids are there. If I talk about secondary, alpha helix and beta pleated sheet, now you can see there is a hydrogen bond. There is a hydrogen bond of, you can say, CONH group of the first loop of amino acid chain with the second loop of, and hence the structure becomes coil structure. Fine. Alpha helix, there are certain saline features, just what you generally remember in case of DNA structure. Such kind of points, don't worry, they are very easy, right? Certain distance that we have to remember, 0.54 nanometer, right? If I talk about right-handed helix, it is more stable, of course, as compared to left-handed, right? Now, when I talk about beta pleated sheet, as I discuss in the structure, that it forms a hydrogen bond between two chain. Fine. Tertiary structure, three-dimensional structure of the protein, of course. If I talk about quaternary structure of protein, more complex one, because now more than one polypeptide chains are definitely involved. Fine. Now, when I talk about frequently asked question, three frontline classification of lipid, amino acids, and protein. Short term of structure of proteins they ask, and Last but not least, that is all about chemical properties of protein and amino acids. Fine. The best book to refer is Biochemistry by Satyanarayan. Fine. So this is all about today's session. In next session, we will discuss about metabolism of all these things. Protein, you can say lipid more specifically. We'll see how they are metabolized. Fine. Thank you. Thank you.